For the end of chapter 5, it talks about cell communication, and the important thing to think about while you're reviewing these videos is that how our cells are going to actually communicate. And so when we look at this, we see that external signals are converted into responses within the cell. So what that means is if you have two cells, so you have a cell right here and a cell right here, and they want to communicate, cell 1 and cell 2, cell 1 can send messages to cell 2. And that's really what we're looking at. So how will cell 2 respond to whatever cell 1 is trying to say to them? When we look at our bodies, we're made out of trillions of cells, and they all work in harmony to have us function. And this relies on messengers inside of our body. So sometimes cells are going to communicate long distances with chemical messengers, and these chemical messengers are our hormones. And sometimes they'll communicate just short distances to cells right nearby. An example of that would be if you have a cut and your skin is healing and repairing, the cells would have to communicate it's time to divide and go through mitosis. Now sometimes if cells have this long distance to travel, they also have that option of communicating right nearby. So there's something called gap junctions in animal cells, that the cytoplasm is actually connected between these proteins found in the membrane, so the ions and different signaling molecules can actually flow right through. Plants have this in plasmodesmata. So here in a plant cell, you can see the cytoplasm and how the cytoplasm is connected between two cells for molecules to pass through. Here's plasmodesmata, and here's plasmodesmata. Now, when we talk about signaling, though, you have local regulators, which is if you have a signaling cell that sends out a molecule, it goes right nearby. Local meaning nearby. So local regulators are for close signaling. Then you also have your long distance. Now, this is where hormones come into play that travel through our bloodstream where they get to their target cells. Now, what you're studying is this. What happens when a hormone attaches to a target cell? Now, the different messengers, the different hormones are specific. You can see these were only specific for the blue hormone, where the yellow one just went right on by. Okay, so now what we're going to look at today also is what happens when that hormone actually attaches to the target cell. Okay, so there's 3 stages. You have reception, which is the hormone or the messenger coming in and attaching to the surface of a protein. Or if it's a lipid, because lipids can cross right through the cell membrane, it'll cross through the cell membrane and actually go in and attach to the DNA or to a protein inside the cell. Then you have transduction, which is bringing about like a cell response. You have what you'll learn, a phosphorylation cascade that occurs. And then the response. Now the responses are super numerous, and we'll see many responses throughout our year. Responses could include the cells dividing during mitosis. It could be gene expression, so turning on or turning off genes. It could be transcription. There's so many cool things you'll learn. So you have your cell response. Okay, so this picture is an overview of the 3 stages. You have your reception, which is when the ligand or the signaling molecule actually attaches to the protein, and that's called reception. Then you have your relay molecules. They're going to pass the message on, and then you have a cell response. Now, the reason you need these relay molecules to pass the message on is because this right here is a protein, and it can't cross the cell membrane because it's not a lipid. Okay, so now let's go ahead and see. So what we're looking at are proteins that are found in our cell membrane. We're looking at integral proteins because these molecules are large molecules that cannot cross the membrane by diffusion. So instead, in order to pass a message on, they need to attach to an integral protein or a membrane protein. So when we look at this, here you have your plasma membrane, and here's your receptor protein. They're going to receive the ligand or receive the message. So in this situation, the ligand's going to come and attach to that membrane protein. and it's going to eventually go through a conformational change. But what we see is that there's 3 different kinds of membrane proteins. You have your G-protein linked receptors, you have tyrosine kinases, and ion channels. Those are all 3 membrane proteins to receive ligands that are protein-based. Now, if it's a lipid, though, because a lipid can cross right through a cell membrane, the lipid has a receptor protein in the cytoplasm or in the nucleus. And these are going to be your steroid-based hormones. Okay, so when we look at an intracellular receptor, here's testosterone. Testosterone can cross right through the cell membrane, attach to an intracellular protein, and then go and directly alter your DNA, turning on protein synthesis. There's your mRNA, there's your new protein that's built. Okay, so now back to plasma membrane receptors, and our first one is a G-protein-linked receptor. So in this one, you have your G-protein-linked receptor. Now your G-protein is inactive, and then you have your enzyme or effector protein. This, when you watch later videos, is adenylyl cyclase. Adenylyl, I don't know how you spell it. Cyclase. Oh, man. Okay, so now here we go. So you have your ligand come in and attach. This is your first messenger. It's the first messenger that the cell has received. The G protein will diffuse across the membrane, and it'll actually become activated in the form of GTP. Now, this is very similar to ATP. Then that ligand will break off, and that G protein will diffuse across the membrane, and it's going to activate this enzyme. It's going to activate adenylyl cyclase. It'll most likely cause a conformational change in shape. But what I just have it change color, but it's actually changing the protein shape, and then that leads to a cell response. So this right here is where you're going to have your second messenger of cyclic ANP, and then you'll have your phosphorylation cascade and the cellular response. But this first part here is how the G protein works. Now, this G protein, though, does not stay attached to adenylyl cyclase. It'll actually go through dephosphorylation and go back to its inactive form. You can see the phosphate comes off here, and now it's ready to go again. If it stays on, that could lead to cellular problems. Okay, so here's a book or textbook picture if you wanted to see what that looked like. Okay, now the other one is receptor tyrosine kinases. Now, tyrosine is an amino acid. So when we look at receptor tyrosine kinases, these are proteins made out of amino acids. We're just showing you here are the tyrosine amino acids as part of this protein. This right here is a protein. Here's its amino acids. And, okay, so let's go ahead and see how these work. So when we look at a receptor tyrosine kinase, you have your signal binding site. Now, kinase is any enzyme that transfers phosphate groups. So they're going to play roles in phosphorylation. So here you're going to have your receptor molecule or your ligands come and attach to your polypeptides, your tyrosine kinases. And when they do that, they actually form a dimer. They come together, and here you have a dimer. And this is an unphosphorylated dimer. Okay, so what will happen, though, these tyrosine regions are now activated. So what's going to happen is here, they'll actually take ATP, and the tyrosine kinases will break off that phosphate, and that phosphate will actually attach to those tyrosine regions. This happens 6 times, and so now you have a phosphorylated dimer, and it's a fully activated tyrosine kinase. Now this fully activated tyrosine kinase, or phosphorylated dimer, is now going to activate relay proteins, and so now when that protein attaches to this region, this relay protein is going to lead to a cell response. Now, when we look at this, you could have like 10, 12, you could have a whole lot of different cellular responses because each of these activated tyrosine regions can activate different relay proteins, leading to numerous cell responses to coordinate events in a cell. Think of something like cell division, for example, mitosis. So many things have to happen in order for that cell to divide. So if a cell receives a message, hey cell, go through mitosis. Okay, it has so many things. It has to break down the nuclear envelope, condense the chromosomes, make copies, divide the cell into two, and that can have multiple cell responses, all because of one signaling event. And here is a picture of that for you. Now, ion channel receptors are probably going to be your favorite, and we see these when we get to our nervous system. This is how our neurons work. So here you have an ion channel that's currently closed, and the ions can't get inside the cell. Well, when a ligand comes and attaches, this could be your neurotransmitter used in the nervous system. It'll actually open an ion gate, and the ions will rush inside. So think of like facilitated diffusion or chemiosmosis. You have all these ions outside, they're going to move from high concentration to low concentration. Now, it could also work where they're moving out. It can go either way, but they're going from high to low. And when you change the charge inside of the cell, that can lead to a cell response. This is critical for proper functioning of our neurons. When we talk about our nervous system, we talk about it as being like sending electrical impulses. Well, it's because of these charges that we consider our nervous system and how it works to be electrical. And this is why we shock people with electricity to jumpstart their immune systems if we need to, if they're going into cardiac arrest or something. And then the ligand can break off, or the neurotransmitter can break off. It'll close that gate. And yeah, that's your ion channel receptors. Now, an example here. So let's say you're in the fight or flight response. So what's happening, let's pretend it's your turn for your verbal test. Epinephrine is like adrenaline, and it'll attach to the G protein receptor, activating the G protein. And then the G protein is going to diffuse across to adenylyl cyclase. And then adenylyl cyclase's job is it's going to activate cyclic AMP. So remember, ATP has those 3 phosphates. Well, what will happen is ATP is going to get converted into cyclic AMP with just 1 phosphate group. So cyclic AMP is now our second messenger. Epinephrine that came in attached was our first messenger. It brought the message, but because it's a protein, it can't cross through the membrane. So how do we get that message to keep on going? Well, we need a second messenger to pass the message on. And so it's kind of like when someone comes to my class and gives me a note to give to a student. So then in that situation, the student, the TA delivering the message is the first messenger. I would be the second messenger that hands it over to the student to go see their counselor or the office or whatever. Now, cyclic AMP is then going to, GDP goes back, and now it's back to inactive. Cyclic AMP acts as our second messenger that's then going to activate the first protein kinase in our phosphorylation cascade. So cyclic AMP that was just made because of that first messenger will actually travel over and activate the first protein kinase, pKa. So protein kinase A is now active, and so protein kinase A is going to activate the next protein kinase. Now remember, a kinase transfers phosphate groups. So here this kinase is active. Ooh, and when it became active, look, it changes shape. So when cyclic AMP goes over and activates it, this protein goes through a conformational change in shape. It changes shape, and that's how it's active. Remember, when we think about enzymes and proteins, how their shape matters. So this would be like an allosteric activator of some kind. So now you have your kinase that's active, and it's going to take a phosphate off of ATP. So here it actually hydrolyzes or breaks apart ATP into ADP and can add a phosphate to the next protein kinase, which will lead to a conformational change, making that one active.